little bit of feedback. I'm not sure if that's normal. Okay. Oh, okay, it's gone. It's still there, but I can manage. So yeah, I was saying thanks, Katya and Martin, for this kind invitation. It's really great to be here and present to you some of our data. This is not my title slide. This is Martin's title slide, but I figured I don't really need to duplicate it. So I will tell you a little bit about the work that are mostly basic work, no in vivo studies for today on the native crack channels and some of the work that we've done in the last few years. And before I delve into that, let me get my pointer here. Most of what I will be presenting today has been done by two PhD students that really work very well together, Scott Emmerich and Ryan Yost. And some of the molecular work was done by my really outstanding technician, Ping, who has been helping the whole lab. And we couldn't have done any of this work without the help of David Yule, Don Gill, and a couple of mathematicians, Vaughan Walter here in Penn State, and Jim Snade, that some of you might know in New Zealand. And we have a list of collaborators here that I will not talk about the work involved in, but I just would like to acknowledge the help of everybody and give credits to all those involved. And so, as you know, calcium signaling is complex. It's an interplay between the plasma membrane, ER, and different organelles. And the pathway that we work on is this tauropated calcium entry pathway that is mediated by crack channels encoded by ORI and STIM proteins. So typically GPCRs or receptor tyrosine kinase. In this case, it's the TCR of the T cell receptor, coupling to an isoform of PLC and IP3 production, causing the calcium release from the ER via the IP3 receptor. And this store depletion is sensed by STIM molecules that have this low affinity EF hand, which then aggregates and move to ER plasma membrane junctions to trap ORI channels and allow calcium entry. And this calcium entry obviously can replenish the stores, but that's probably not the most important function, because perhaps every pathway can replenish the stores, but it's mostly transcription, and namely the activation of NFAT isoforms. And so, as all of you know, there's no crystal structure or cryo-EM structure, at least not that I know of mammalian ORI. But the Drosophila ORI crystal structure has been solved in 2012, and it's a hexamer. This is the monomer here with the TM1, and that's the selectivity filter. That's the human equivalent E106, which is right on the outside here of TM1. And you can see the six amino acids that coordinates the calcium ion just right here. So this is the choreography of crack, and what's known and what's been focused on. And I do not want to mention any names just by fear that I will forget some of the names of the people that have really contributed amazing work into the understanding of the details, the atomic level details of the unfolding of STIM, the SOR region or CAD with its CC2, CC3 here, and the interaction or the interaction of this SOR domain with the C terminus of ORI channels to allow calcium entry. This is a slide from the Romanen lab, but I may as well use any slide from any other lab, because you probably saw variation of this cartoon from many different reviews by many people in the field. But what I'm trying to say here is that the studies have focused mainly in this interaction between STIM1 and ORI1, and that's for a good reason, because they are the major player of crack channel and tauropated calcium entry. But one of the things that we have been interested in is this different isoforms of ORI. And just to remind you, that although invertebrates have one ORI isoform, vertebrates have two, and in mammals you have three. ORI3 seems to be appearing in mammals from apparently duplicated from the ORI1 gene. And so this begs the question: is why do mammals have these different isoforms, and if only STIM1 and ORI1 can mediate crack channel activity? And at first, one might argue that this isoform mediates crack channel activity in specialized cells of mammalian systems, but that actually turned out not to be the case. And if I attract your attention to the pioneering studies by Stefan, Yusang, Morali, Rich, Patrick, and Angela, or by Monica, Reinhold, Jean-Pierre, or Mike Cahalan's group, ORI2 and ORI3 actually did not give a hit in these screens. It was only ORI1, and I think this paper fished ORI2 and ORI3 by homology from ORI1. So by themselves, they didn't really contribute an appreciable or amount of crack channel activity or SOC as measured by fluorescent probes. And so this is the question that has really bugged us for all these years: is that what do this ORI2 and ORI3 really contribute in this regard? And a way to really tackle this is just to do this systematic study where we go in and we knock out every single isoform alone or together. And just to, before I delve into this data here, I'll let you look at it while I am explaining what we've done. We just took all the 5 genes, STIM1, STIM2, ORI1, ORI2, ORI3, and we've done any imaginable combination you can think of to knock them as a single, double, triple, quadruple, and look and generate several clones by independent guides, CRISPR-Cas9 guides, not just clones like using the same guides, just to eliminate any kind of off-target effects. And then we did I can skip all the details because you can look at some of the data in this paper. We did all the sequencing, the Westerns, the qPCRs, and in most cases we studied 2 clones per, sometimes 3. And this is the result by using this tepsigargin protocol: calcium off, adding tepsigargin in the absence of extracellular calcium, then restoring 2 millimolar calcium here. And not really very surprising, as when you knock ORI1 and SOC activity is essentially gone. You might see that there is some tiny minuscule level of SOC activity here, but I think you'll agree that it's pretty much gone. But then when you look at what you get with ORI2 and ORI3, it's a little bit different. And with ORI2, you have an enhancement of SOC activity. And even with ORI3, you do. This is one clone that has brought this down, which is not kind of statistically significant. But as you will see from subsequent data that I'm going to show you, and as you see here from the double knockout, you can see that this effect of the double knockout is more pronounced than the single alone. And if you look at again, ORI1-3 or 1-2, as long as you remove ORI1, again, you essentially have very little or no SOC activity. You can, if you kind of squint your eyes, you could see that with ORI2 alone left here, it's a little bit more than ORI3 left alone. And with both of them, maybe it's a little bit more, but that's just requiring a lot of faith, not really statistical evidence. And that's just the composite of the data. These traces here are based on hundreds of cells. And you can see the data here with ORI1 and 1-3 and 1-2 knockouts giving you nothing. Just before I move on, I just would like to give credit here to actually work that has been done, and this we are not claiming that we're the first one to do that, but there's a beautiful paper by Stefan on ORI2 in CD4 T cells. There's another excellent paper by Mark Freischel on mast cells, and another paper by Rodrigo La Cruz on enamel cells, showing that this is essentially the case that if you get rid of ORI2, you can enhance SOC activated by tepsigargin, suggesting that these molecules are negative regulators of crack channel activity. And because crack channel is a hexamer and it's 6 subunits, then the idea is that they do this through heteromultimerization, right? And again, this is not something that is not known. This is known, has been described before, but this is our own experiments, our own recordings of looking at the calcium-dependent inactivation of all the 3 ORIs. Those are all ORI3, ORI2, ORI1 expressed with STIM1 in ORI triple negative cells. So there is no contribution from endogenous ORI. And where we look, normalize to the fluorescence and record from cells that have equivalent level of fluorescence, because all the ORIs are tagged with YFP. You can see here in this voltage step protocol here that the peak current here and the inactivation in this 150 millisecond step, that I hope you could see it, that the inactivation of ORI2 is more pronounced than that of ORI1, and the inactivation of ORI3 is more pronounced than that of ORI2. So you have, when you look at current density, you get peak current, you can see this ladder. And that's the data here. This is the remaining currents when normalized to peak current, taken about 3 or 4 milliseconds here before the end of the step. You can see that the remaining currents, there's much more remaining for ORI1 than for ORI2 than for ORI3. And so these channels have a different, on their own, clearly they can form crack channels. It's been known. They can mediate crack channel activity, calcium entry, but ORI3 is not as good as ORI2, and ORI2 is not as good as ORI1. Okay, and so just to compare this, this is the single ORI here. That's from this data here. And if you compare it to when you co-express 2 ORIs at the same time, again in the triple knockout, 1 and 2, 1 and 3, or 2 and 3, or if we expressed here a tandem of 1, 2, 1, 3, 2, 3. I hope you could see easily again here from the current density and just from the traces here that you generate somewhat of a hybrid kind of inactivation profile. And then the peak current is also altered accordingly, suggesting that these channels, if they heteromultimerize, they generate channel activities that are somewhat in between the two, or at least dampen them. And this could explain how ORI2 and ORI3 mediates crack channel, ORI2 and ORI3 mediate inhibition of ORI1. So I don't, we have some other evidence based on the probing the native with 2-APB. You can go and look at that in this 2020 paper that we just published in Nature Communications. But so far, what does all this mean? Okay, you have ORI2 and ORI3 inhibiting ORI1. Okay, but inhibiting how and why, and you add in, you maximally activating SOC and okay, and you're suggesting that it's probably inhibiting right here when you have maximal activation of SOC activity or crack activity. But in essence, this maximal activation as measured by tepsigargin or with patch clamp and BAPTA in the pipette is probably just one aspect or one window of this vast array of calcium responses that are generated by mammalian cells. And this is, again, not something that I'm teaching this audience. You guys know this very well, and you're familiar with the oscillation models and how this, with increasing concentration of agonists, you can have this increase in frequency of calcium oscillations until you just reach these plateaus. And so I hope I could tell you that that's what we set up to do, and we were really very clever, and we thought, oh, well, we decided to look at these ORIs in the oscillations, but really that's not how we did it. It's sometimes, not sometimes, all the time, it's better to be lucky than smart. And what we did is that we made an observation, which I will get to in the next slide, that put us in the right track. But for now, I can tell you that if you look at the under this oscillation protocol, this is using 10 micromolar carbachol. You can see it here. In every slide, I try to put the concentration that we used. And you look at, this is 5 representative cells of the wild type and 5 representative cells of the triple knockouts. This is 1, 2, 3 triple knockouts. And here you get a combination of oscillations. You get some cells that plateau, but by and large, most cells oscillate because the concentration is relatively low. But with the triple knockout, you get the typical 1 or 2 or at most 3 oscillations, and then the cells cease to oscillate. After that, there is nothing. And you could see it here in the quantification of data from a lot of different cells from different runs. So in essence, if you look at a pharmacological inhibition of calcium entry, or by any other means, by any drugs or by gadolinium, this is how it looks like. So it looks just exactly like the triple knockout. So what really put us in the track is this experiment right here. And that's when we realized that something is off, something is not really right. And we've dotted a lot of I's and crossed a lot of T's to be absolutely 100% confident that this data is real. It's not off-target effects, it's not an artifact. And that is that the ORI1 knockout, the one that are missing essentially all SOC activity, continue to oscillate unabated, undisturbed, even for over an hour or more. And let me just attract your attention just to these 2 panels here, this one and this one. You could look at all these because that's the different combination of knockouts with the representative oscillations. But what you could see here, if you look at oscillating cells versus cells that show response in the form of plateau, this is at a relatively low concentration of 10 micromolar carbachol, you would see that with ORI1, not only they oscillate just fine, but it's kind of slightly even a higher number of cells that oscillate. But what I would like to attract your attention is to, again, this ladder phenomena here. When you go to ORI2 single KO, the oscillating cells, the percentage of oscillating cells decreases, more so in the ORI3 single KO and more so in the double KO. However, on the other hand, you could see that this percentage of cells that show your response in the form of plateau follows the opposite trend. That is, the increase in ORI2 knockout, even more in ORI3 knockout, and even more so in the double knockout. Maybe not significantly, but there is a trend there. That's the first thing. The second thing is that notice here how the ORI1 single knockout, or for that matter, any knockouts that doesn't have ORI1, 1-3, 1-2 double knockout, there is not a single one, none, zero cells in every single recording that will show you a plateau. So this basically says that ORI1 is the major subunit, and by itself it can mediate these plateaus fine, but it's really ORI2 and ORI3 that tailor the calcium responses to the level of agonists. That's how the negative regulation by ORI1 of ORI1 by ORI2 and ORI3 makes sense. You can see it in the representative traces here. With ORI2 knockout, you start seeing more plateaus, more so with ORI3, and then with ORI2/3 at 10 micromolar carbachol, you essentially have 80% of the cells that are responding with a plateau. So now think about the consequence of this, is that a cell is encountering a concentration of agonists that normally would make 80% of the cells oscillate. But now the cell is thinking, because it doesn't have ORI2 and ORI3, it's thinking somehow that that's really a maximal concentration of agonist. And it's most of the cells are plateauing. And that's what the role of ORI2 and ORI3 is, to dampen the activity of ORI1. Not only to play a role at one specific concentration level, but across the range of all concentrations. So all these channels are most likely involved from the very beginning to the bitter end, and they are involved. Now, what's the stoichiometry? What's the oligomeric states? That's something I really don't know, and that's something that we are really interested in knowing. And so let me drive this point home a little bit further and show you this recording where I focused here just on the ORI1 knockout and ORI2/3 knockout, double knockout. You can go to the paper and you see all the other combinations there, but just for the sake of time and to convey the message more clearly, to show you this. And if you look here, this is an increase in concentration of agonists on the same cells. You go from 1 to 3 to 10 to 30. We go all the way to 90 or 100. But in this recording, we stop at 30. And you can see that the more you go, you start oscillating, and then the oscillations get bigger, and and then some cells plateau. But with the ORI1, that never happens. They just continue to oscillate. But with the ORI2/3, not only did a lot of them plateau, but notice how at lower concentration of the agonist, they already show some responses. But certainly at 3 micromolar, you could see that all of them already respond. Even some of them showing a plateau at 3 micromolar. So again, they become more sensitive. You could see the data here again. Look just again at the teal, which is the double knockout, and look at the red, which is the single knockout. Look at this trend. You go down here with the concentration of agonists, and you go kind of up here and plateau for the red. But then it's the opposite here for the plateau cells. They keep going back up, and obviously they meet the, you saturate here and you meet the similar level to the wild type. But then, because you don't have ORI1, there is absolutely no plateaus for these cells. Okay, so James Snade really was very helpful, and he plugged in all this experimental data with all the model of the oscillations that has been worked on and before, especially with David, and considering just association between the different ORIs as dimers, just to simplify the math. Here modeling only ORI2, assuming that ORI3 is about the same. And this is what the model gives with the tapsigargan, is that ORI2 is a negative regulator, same for ORI3, and that the two together work even better. And all the oscillations kind of matches. The model reproduces the experimental data with these 3 caveats, which 2 of them we definitely verified experimentally. And these are the first 2. Is that the first one is that for this to work, ORI2 and ORI3 should allow less calcium current than ORI1, which is, we know it's true. It's experimentally documented by, not us, by many other people. And that ORI2 and ORI3 relative proportion in native cells are 20 to 50% of this of ORI1. And that's something that we documented experimentally using knock-ins and using like a fluorescence to show that indeed most likely ORI2 and ORI3 proportions are less than that of ORI1. And the third assumption is that ORI2 and ORI3 more readily associate with ORI1 than with themselves, and that's something that we have not verified experimentally. What does this mean to NFAT translocation and to the transcription? Well, looking at the reporter NFAT GFP, NFAT1 GFP, NFAT4 GFP, let me just start here by just the two windows here. The top with NFAT1, and you can see this is, look at the time scale. This is 30 minutes. This is 15 minutes. You could clearly see that in the wild type cells, NFAT1 moves slowly, more rapidly for NFAT4, but clearly for the triple knockout, nothing really moves. Well, however, here, if you just look at the double knockout ORI2 and ORI3, I hope you could see clearly how this translocation of NFAT1 and NFAT4 is huge, bigger. But one thing that is really more important that I want to attract your attention to, look now how these 2 curves for NFAT4 and NFAT4 on NFAT1, they almost look the same. I mean, by 15 minutes here, you almost have maximal translocation, which is about the same thing as here, maybe a little bit more. So again, because you've skewed the calcium signal by removing ORI2 and ORI3, the cell has no way of distinguishing even between the microdomain of calcium that is required to differentially translocate NFAT1 versus NFAT4. And that's where that is important. So in the next 5 minutes or so, 5-10 minutes or so, let me just tell you now how we try to put all this together within this big picture of all the STIMs and all the ORIs. And that's what we did. This is work that is currently under review that I'm going to share with you because it's unpublished. And that we did these combinations again of different STIMs and then different STIMs with the ORIs. And this is just documenting the knockdown of STIM1, that of STIM2, the double, missing both. And again, just like with the first paper, we looked at all the IP3 receptors to see any compensation, the buffering, the replenishing. This is using the IP3 receptor triple knockout from David Yule and his antibodies. And not expected here. If you look at the Tapsigargan, you could see what's been published before. With STIM2, you reduce the SOC activity a little bit. With STIM1, you reduce it a lot. With double STIM1, STIM2, you completely abrogate it. And we're showing here the triple ORI knockout, just to show that the triple ORI knockout essentially phenocopies in these protocols, phenocopies the double STIM knockout, which is totally expected. But now, let me take you to the oscillations, and this is with 10 micromolar carbachol. And one thing you would notice is that with this concentration of agonists, the cells, whether you get rid of ORI1, STIM1 or STIM2, the cells will continue to oscillate. However, there is a slight difference, is that with STIM2, well, again, more cells will have a tendency to show you a plateau. And you could see it much more clearly here in this protocol where you apply increasing concentration of agonists. You can see that with STIM1 knockout, they will never plateau again, because that's what is needed for them to plateau is STIM1 and ORI1. But then with STIM2, notice now how a lot of cells start showing a plateau a lot earlier, while the wild type haven't. And that's, again, the data here by focusing on oscillating cells versus plateau cell. Look, just compare the blue to the red. The blue goes down, the red goes up and tapers off. The red never moves from the ground. The blue keeps going up. And that's, again, suggests that this model that STIM2 is working under homeostatic condition, then STIM1 kicks in when the agonist is there, is wrong. It's not correct. They all working together all the time, from the very, very beginning to the very end. And some of the data from Indo Ambudkar, I think, suggesting that STIM2 can drug ORI1 is definitely in agreement with these findings. And now, here's another observation that was totally unexpected, and that is the behavior of the STIM1/2 double knockouts. This data is based on several clones, and this is again, I just want to make a parenthesis here. This is one of these cases where these differences, siRNA would have never been able to resolve this, especially this negative regulation of ORI2 and ORI3. There's no way siRNA would have been able. So that's the kind of, every technique has its cons and pros, and this is perhaps one of the strengths of CRISPR-Cas9. But anyway, let me get back to STIM1, STIM2 knockout and what you see here, which was totally unexpected. You add this 10 micromolar carbachol, and what we have is this, like a stream of oscillation, and then just dying out, very fast. Just contrast that with the triple knockout ORI, which in the Tapsigargan protocol, they phenocopy each other. In the oscillation protocol, not so much. So why is that? This is clearly not related to calcium entry. It's related to something else. So we thought this is probably a regulation of the IP3 receptor. You could see it more clearly here with this increase in concentration of agonists. You could see here they're more sensitive. Look at 3 micromolar, they're already spiking more so than the wild type. And then by 10 micromolar, they've already spilled their gut. Then after that, it's just they really have nothing to give. They're done. And so what it turns out is when we do direct ER measurements with sepia, and then we use this maximal concentration of carbachol in the absence of zero calcium, and then we remove carbachol and we put calcium to replenish. Let's focus on the replenishments. Not unexpected. The double knockouts flat. The red here is the STIM1 knockout, seriously slowed, marginally slowed for the STIM2 knockout. But look at the depletion. This is where we notice that if you look at hundreds of cells again, you look at the depletion, you see that with STIM1 and STIM2 knockout, there is some maybe fast depletion, but it's not significant, definitely not, but it's definitely significant for the double knockout. And you can see the data here, the quantification of the data here. And that's just the quantification of the refilling. So this suggests that the STIM molecules are doing something to ER, and they are somehow slowing the depletion. And when they are not there, the depletion occurs very quickly. And just before you think that this might not be related to STIMs, we kind of rescued that. And so if you rescue, you can rescue. So this is the rescue. We use this this weak promoter, this thymidine kinase promoter, to just put amount. This is the rescue amount. This is the wild type amount for STIM1. Same here for STIM2, rescue amount and wild type. You can see the Tapsigargan protocol, how STIM1 rescue SOC, STIM2, again, just maybe a little bit higher than what native STIM2 does. It's really very hard to dial, but you get the idea. And the oscillations now are recovered. They are more spaced out. They are not like this. And before I show you the ER measurements for this experiment, let me point out, when we look at chimeras, where we use the C-terminus of STIM2 with the N-terminus of STIM1, again rescued with this weak promoter, or the opposite, the C-terminus of STIM1 with the N-terminus of STIM2. You can see with the Tapsigargan, it's the C-terminus really that drives calcium entry via SOC, because the green trace has the highest, and so it matches what the C-terminus is. And here the blue trace, which is the STIM2 C-terminus, it just looks kind of like STIM2. So it's the C-termini, and in terms of oscillations, they match what C-termini of STIM1 matches STIM1, the C-termini of STIM2 matches STIM2, and it's the same in terms of rescue of the oscillations. This is looking at the STIM1, STIM2, and this is at the C-termini within the chimeras with the N-terminus of STIM1. And so it's the C-termini that actually determine the oscillatory profile and how STIM2 can be a negative regulator of STIM1. But what about the N-terminus? Not the N-terminus, but what about the N-terminus? So now I go back to this behavior here, and to show you here how this behavior is by looking at direct ER measurements with sepia. And there is no easy way to do this, because I don't really want to get into a rabbit hole here. But if you use EF hand mutants, they're going to be constitutively active, as all of you know. So what we did, we used this STIM2 variants and STIM1 variants that basically have non-functional source, so they cannot activate ORA channels. This is the STIM2.1 splice variant that was co-discovered by Barbara Niemeyer and Rich Lewis, and this is the F394H mutation that Don Gill kindly gave us, that basically does not activate ORI, and if you now have nothing to do with entry, you rescue with these two variants that really do not mediate store-operated calcium entry. What do you get? You get this shift back, where now these guys alone give you a little shift, maybe not significant for STIM2.1, but significant for STIM1 F2H mutation. But together, the shift is more significant. Now they are slowing down the release in response to carbachol. And what else is that now they're spreading the oscillations more, and together they're spreading them even more than alone. And the reason why they run out is just because these are non-functional, so there is no calcium entry. So eventually they will run out. You expect them to run out, but I hope you could see that they can extend this release. And now when you go to the N-terminus and you mutate the EF hand, they don't. So this is the EF hand within the F394H. This is the EF hand mutation within STIM 2.1, and that are the combined, and they look exactly like STIM 2, like STIM 1-2 double knockout. This suggests that it is the N-terminus EF hand. It's something that keeps STIM inactivated. That's what negatively inhibits calcium release. And to provide some evidence that this is occurring via the IP3 receptor, that's where Dave Ewell helped us with some occasion experiments. He did, Vikas in his lab did carbachol, addition of carbachol to these cells, but also occasion of IP3. I hope you could see here this this is IP3 receptor activity measured in turf space, that STIM 1-2 double knockouts have more IP3 receptor activity. You can see that the number of puffs and the number of puff sites are increased, and the number of cells that show like a globalized calcium signal within the first 20 seconds or the first 60 seconds is definitely increased. This is just telling you again this more touchy-feely behavior of the STIM 1-2 double knockouts. However, the time of rise and decay of the peaks or distribution of the amplitude did not change, suggesting that perhaps the fundamental biophysical properties of IP3 receptor is not really affected. It's mostly the number of puffs and the number of puff sites. And so the model totally reproduces this, and it actually, for the model to work, you have to have a cooperation between STIM 1-2 with only STIM 1 will give you new plateaus. If you get rid of STIM 1, you can get the plateaus. And then the unactivated STIM 1, the resting STIM 1, somehow indirectly, we know because they don't co-IP, inactivates the IP3 receptor. And you can see that when you remove 1 and 2, you get the speeding of the oscillations and then nothing. And even the ER depletion qualitatively reproduces the data that they are, if you get rid of STIM 1 and STIM 2, the calcium release is enhanced. And so the model suggests that at rest, 7% of STIM ORI mediated calcium current is mediated by STIM, and the remaining 93% is by STIM 2. And at maximal concentration, the table is reversed. But in between, it's a continuum. As the contribution of STIM 1 increases, that of STIM 2 decreases. Again, they work together. They cooperate and they work together. And that as the inactivated STIM negatively regulated IP3 receptor, as perhaps setting a threshold for IP3 to activate IP3 receptor. And as STIM become activated, the negative regulation is lifted, and by the same token, ORI are activated. And a final thought is that because look, even the double knockout, if you do the ER measurements, they start here. They have their ER4, and that's something that all of you know. And that's actually, it's kind of interesting because the model predicts that there must be a pathway at rest that could mediate this refill, that at rest only 28% of the calcium influx is through STIM and ORI channel, and the rest is through this mysterious pathway, which I don't really know what it is, and that this increases to 92% at maximal agonist concentration. And so, final slide, I promise, is how all this translates to in-fat nuclear activation, and this is where I could have never predicted this, and siRNA could have never given us this clear result, and that is look at this low concentration of 10 micromolar. This is the high 300 micromolar. And whether you knock out STIM2 alone, STIM1 alone, or STIM1, STIM2, not much happens for the translocation of NFAT1. For NFAT2, there is some translocation, but you could see that clearly with both of them, it's affected. At higher concentration of agonists, STIM1 is clearly playing a much more prominent role. Read here, because this is knockout of STIM2. You have only STIM1 here in the blue trace, especially for NFAT1. But when you look at NFAT4, both of them are kind of equally contributing to this translocation. This is again that there's this cooperation between the two across the range of agonist concentration. There's this choreography. This it's not just as simple as yeah, they don't have a redundant function. One of them plays here, the other one plays there. They just work together. And I don't know if they form heterodimers or two homodimers. I think that's something that remains to be seen. And so finally, at the horror of my Latino friends or Spanish friends and me, I using this danza de los cinco. My Spanish pronunciation is probably not what it is, but we should probably view this pathway not just from the standpoint of just STIM1 ORI1 or STIM2 ORI1, but all the five, involving this intricate dance with the IP3 receptors. And that if you have only STIM1 and ORI1, you probably have just like an on and off switch. But if you consider all the five, then you could dial this various amount of frequencies and calcium signals, and then you can actually distinguish between different activation of different isoform of transcription factors, and you could account for this mammalian complexity of the calcium signal and the fidelity of the calcium signal, where every isoform is translocated at its intended calcium microdomain. And another thought, and that is that this crack channel, native crack channel, should be considered as a heteromultimer of all these. And again, I cannot say this is just a cartoon. The stoichiometry remained to be determined. But what is clear is that that this channel, if you with ORI2 and ORI3, definitely allows less calcium than with ORI2, which allows less calcium than with ORI3. And I think the contribution of STIM represented here by a pie chart, where one of them, STIM2, starts high and then changes, and this communication with IP3 receptor, where I think this really ensures that also the amount of IP3 receptor function or calcium release coincides with how much IP3 production is and how much store depletion is, and is dialed into the proper amount of calcium entry. So all these molecules are constantly in dialogue with each other to coordinate the calcium signal and ensure the fidelity of the complex mammalian calcium signaling. And that's my final slide, and I'm happy to answer any question you might have. Thank you for your kind attention.
Thanks a lot, Mo. We have some questions, so it would be great. Now I will go on with the questions. So it would be great if we can answer them relatively quickly so that we can cover all of them, and we are still in the time. So Jonathan had one question, but I think you answered that in the next slide. So I will start with the question from Alex Simpson. Alex would like to know, does the store content, the ER store content, differ in ORI knockout cells?
Quick question. All of them have perfectly fine stores. I attract your attention to this paper in Flugers, Alec, by Eugene Wong, who did really measurements and compared all this. And essentially, the double knockout STIM or the triple knockout have only marginally decreased ER store. I don't remember the numbers off the top of my head, but 85 to 90% full. But they essentially, all of them are essentially full. They are not really what you would expect to be completely depleted, you know.
Okay, thanks, Mo. Then the next question by Agnieszka Dirda. How other type of channels, for example, TRPCs, could affect or impact these calcium oscillations?
Yeah, no doubt. I mean, they could, and there are native TRPCs expressed in these cells, and TRP in general may be some of some of the channels that could replenish the stores. And this is actually, there's some earlier studies by Jim that if you put TRPC3, then TRPC3 will refill the stores, no problem, you know. So that's a short answer, but you know, but I don't really know more than that.
Okay, thanks. Then the next question comes from Martin Butman. So the question is, are STIM1 or STIM2 regulating calcium release at the ER and or PLC activity at the membrane? And would this altered IP3 production explain the effect of STIM1 and STIM2 knockout?
Well, the data suggests, I mean, if you trust the data of IP3 engaging and the fact that we've done all these controls to show that the machinery is not really that much altered in those knockouts. So we don't think it's just because it's a shift in the agonist sensitivity or PLC activity or changes in IP3. It's something that the stims do to the IP3 receptor, and we know it's not direct. So we know it's not a direct binding because actually David did try some co-IPs between stims and IP3 receptors, and there is no evidence that they interact. So we think it's a regulation of the IP3 receptor. It's just not a direct one. I don't know how. That's just a mystery to us.
Okay, so we have some related questions. Sandeep Patel asks if ORI2 or ORI3 knockout affects the store release or IP3 like STIM knockout.
Yeah, no, not at all, not at all. And again, you know, when you look at when you measure directly the ER, you know, you don't see, actually for single knockout, you don't see any difference. Once you start dealing with the triple knockout ORIs and the triple and the double knockout STIM, there is some differences in the refilling of the ER, like I explained in response to the first question. But as in terms like of oscillations and IP3 mediated calcium release, we didn't see any difference. They don't affect any of that. We don't think so.
Thanks. So you answered the next question with that. So I would go to the question by Neha Gupta. Do we know if in the absence of two ORIs, the third ORI is overexpressing in order to compensate?
Absolutely, yeah, absolutely not. We looked at that, and if it was overexpressing to overcompensate, we would have seen changes in the pattern. I mean, I can go on all the details with the experiments we've done, but just for the sake of time, evidence on changes in expression, in profiling, in calcium measurements in the ER and the cytosol suggests that's no.
Okay. And another connected question. So the same thing about IP3 receptors and Zerka. Do you see some difference in expression, Mo, when you?
Yeah, absolutely. Yeah, absolutely not. Absolutely not. And even further than that, functionally, if you use this gadolinium insulation, and you just basically take the calcium entry out of the equation and allow just the cells to continue to oscillate without any involvement of calcium entry, you could see that the amplitude and the frequency of the calcium signal between all these things is maintained. And so there is no changes. And then by Western's, you could see that there is, I didn't show the data for the ORIs, but it's in the paper. There is absolutely no changes in all the three isoforms of the IP3 receptor. So it cannot be explained just by that.
Okay. The next question is by John Sobolov. So Jonathan asks, so it seems like ORI2 and ORI3 are serving simply as negative regulators of ORI1. If this is so, would expressing less ORI1 have the same effect as overexpressing ORI2 or ORI3?
I can only, we haven't done the experiments, but I can only assume so. And when Jonathan says simply, I mean, yeah, simply, but it's a pretty important thing because if you look at all this evidence that we have from primary cells and how this changes, this remodeling of expression of all these different ORIs, look at the work by Stefan on T cells and how changes in ORI2 expression can control the extent of crack in effector T cells versus naive T cells, or look at the remodeling of these ORI channels in cancer cells and in different diseases. So clearly, by changing just the amount of each ORI, you can dial the calcium signal to whatever this I know it's a teleological argument, but to whatever the intent is. And so yeah, I think it's pretty important.
Okay, thanks. I guess you partially answered the next question that Jonathan had, but I will go now to the next one. So what about the eye crack? How does it look at the plateau scenario or at the oscillation scenario? So that's a question by
Zhang.
Yeah.
Well, I can tell you how it looks. How it looks when we measured it by including BAPTA in the patch pipette, and that's the native eye crack. But trying to measure eye crack under conditions where calcium is buffered inside the cell and where you looking for crack channel activity under a very low concentration of agonists, that's not something that is easy to do, and we haven't even attempted to do that because I don't think, I mean as it is in HEC, and a lot of people, as it is in HEC, a native crack, you're dealing with something like 0.2 to 0.5 picoamp per picofarad when you're using 20 millimolar BAPTA to prevent calcium dependent inactivation and to maximally deplete the stores. So I don't think it will be something that you could expect to see at such low concentrations of agonists.
Okay, Mohamed, one question now from, just a second, Catherine Leclerc. Does this mechanism can be extended to other cell type other than HEC cells? And maybe there I would add also a question from me. I always remember these papers by Ricardo Dolmetsch and Don and Jonathan Sobolov, where 2010 it was shown that STIM actually inhibits L-type calcium channels. So can this regulation be kind of integrated in the nice model that you presented, or we see now in this last slide?
Well, I would think so, because if you look at the negative regulation in the different primary cells that I told you about, from Rodrigo, from Mark Freischel, from Stefan, of course they haven't. I don't think so. I hope I'm not misquoting their own work. I don't think they've looked at various concentration of agonists and increasing concentration of agonists. But it's clearly that those are primary cells and different cell types where ORI2 is negatively regulating crack channel activity. So I would assume so. I don't have any experimental evidence for it, but I would just add that the level again of expression of ORI2 versus ORI3, or just ORI2 or just ORI3 with ORI1. And certainly, we do think, based on some of the work done in my lab, that there are cell types where they will have ORI1 and 3, but not much of ORI2, and vice versa. And so that's possible, but I don't have any evidence for it. The second question regarding the negative regulation of L-type calcium channel by STIM again, I can just do some hand waving. I would assume so, but I have no evidence for it, none whatsoever.
Okay. Thanks. So I have now an interesting question, which I try to simplify, by Jason Bruce, and it is, what about ARC?
Yeah,
You know what
a very
is
good.
meant?
Yeah, very good question. So this is something that I thought about a lot, and you know, if you look at the ARC data and all the stuff that has been done by Trevor and few others, one of the major premise is that there is no store depletion, and so just think about the involvement of a heteromultimer of ORI1 and ORI3 under conditions where you would have almost undetectable levels of store depletion. Could that explain the ARC data, and could lipids or whatever just serve as things that sensitize this channel, and that's basically what it is? Maybe. Could it be its own separate entity? I don't know. I think it's too early to tell. I'm just keeping an open mind about that. But it did cross my mind that you could look at that single ORI1 knockout with the Tapsigargan protocol, how you can't even see much of with calcium entry with that protocol, but the oscillations are continuing strong, suggesting that you really do not need much to sustain the oscillations. Something that the dyes cannot really measure, and yet it's enough to refill the stores, to support oscillations just fine. And that's exactly what the ARC model says.
Okay, thanks, Mo. We have two more questions. I guess we can finish them. The next one is by Kevin Foskett. So Kevin asks, what about STIM1 and possible interaction with annexin A1? Because
yeah. So that's a very important point, you know, because of course I know Kevin's paper very, very well. I read it very carefully, and that is something that definitely did cross our mind, and it is in the works, definitely. So because his data shows this luminal regulation of the IP3 receptor by the annexin, and that's a calcium binding protein, and so yeah, it's just out there begging to be done. Yeah, very good point.
Okay, Mo. So we have one more question. Rajesh Bardwaj wants to know if you have considered looking at these effects in MCF7 cells, because you have worked with those cells before.
Yeah, that's a good point. So there's one. So we haven't done it. Not that we don't want to do it. We haven't done it. It's just because it's a lot easier to do CRISPR and HEC than to do it in MCF7. And so at some stage, what we would like to do is look at the knockout of. We did the knockout of ORI1 in MCF7. And this is some of the work we've done in collaboration with Greg Monteith in Australia. And it's in the works that we will do the double knockout for ORI3 and ORI1 in MCF7. That'll be really interesting to see what we find. Because in MCF7, again, those are cancer cells. And it seems that the ORI3 alone is doing a lot more than the ORI3 is doing in HEC, because based on the 2APB data, 2APB activation of ORI3 and the knockdown data, the calcium with the Tapsigargan protocol is significantly reduced, suggesting that the picture there in those cancer cells might be completely different than in cells like HEC or in primary cells from other origins.
Second, Mo.
Yeah, I'm here.
So yeah, just my microphone was off. Thanks a lot. We don't have any other questions. I do have some, but I can ask you later. We don't want to make it too. Yeah. So it was really great talk, and
thank you.
A great discussion. I want to thank you for your really very, very exciting and nice presentation. But I also want to thank Katja and Martin for organizing this. It's really great to see all the people here. Yeah, because we need this now, and I hope that soon we will, see each other at some nice location, and we can share some new data and have some fun, of course. Yeah. So I wish everybody a nice day, nice evening, and see you soon. So.
Yeah. Thanks everybody. Thanks for listening. Thanks Katya and Martin and Ivan, and to everyone who was here.
Thanks very much, Mo. Great talk. Really good discussion. Thanks, Ivan, for moderating. It's definitely a talk I'm going to go back through the recording of. It was great fun. Lots to take in. If you want to stay on and anybody has any questions, more than welcome at this point to open their microphones and ask questions if they want. If not,